Hello all, welcome back. In this lecture, we're going to talk about evolution. What I'm going to do is I'm going to give you kind of a broad overview in which we look at the mechanisms that drive evolution, and then we're also going to look at a couple of examples. So let's begin with a basic definition. Evolution is change over time. And when we talk about this change in terms of biology, the type of change that we're talking about are heritable changes. Specifically, the changes that we see are mutations. Now, there are four mechanisms of evolution, and none of these work in isolation. Oftentimes, to create evolutionary change, these all work together. Mutations are one of those mechanisms. So let's take a couple of minutes to briefly review mutations and how they may affect this evolutionary change. You'll recall that mutations are changes to DNA sequence. So there are three different types. There are silent, missense, and nonsense mutations. Silent mutations have no effect on protein sequence, even though they do change DNA sequence. Missense and nonsense mutations are going to be those that will change an amino acid sequence, and so those are going to lead to changes in the structure and the function of proteins. So for example, if our DNA sequence shown here has the sequence 3' TCC, ACA, GGC, and if this is template DNA, we know that the mRNA is going to be complementary and anti-parallel. And so the sequence would be 5' AGG, UGU, CCG, where T's in DNA are paired with A's in mRNA, C's are paired with G's, G's paired with C's, and A's in DNA are then paired with U's in the mRNA. Recall, mRNAs do not have any thymine or T nucleotides. This mRNA then, if we were to take this 9 nucleotide sequence and divide it into 3 codons, we would get the protein sequence arginine, cysteine, proline, or ARG-cis-Pro. Let's imagine that there's a mutation though. And so if this fourth nucleotide, which is an adenine, is mutated into a T, what we would find is that this would alter the mRNA sequence, and instead of the codon UGU, we now have AGU. And if you were to use a codon code, what you would find is that this codon codes for arginine instead of cysteine. And so mutations like this are going to change the structure and the function of proteins. Some mutations may have more dramatic effects on structure and function than others do. But it's important to note that these mutations can be passed down from one generation to the next through the process of mitosis or through the process of meiosis. If these mutations are going to be passed down from one generation to the next by meiosis, these mutations have to occur in the germ cells that develop into sperm or eggs. Let's now talk a little bit about natural selection. I'm not going to get into the historical framework of this other than to just point out that it was Charles Darwin who first proposed the theory of natural selection, though let's be sure to note that he was not the only one that had developed this theory or was working on this theory. In fact, there were many other scientists at the time that were looking at how humans had artificially selected organisms, for example, how we breed dogs for certain traits, or how we breed crops for certain traits like corn for bigger ears or bigger yield. Those were examples that kind of drove this theory of natural selection, whereby it's not humans who are selecting, but it's the environment that is selecting organisms based on certain traits. And so natural selection is a theory in which organisms that are better adapted to their environment will survive and reproduce. And that term survive and reproduce, we call that fitness. There are basically 5 guiding principles to natural selection, or that are needed for this to occur. The first is there has to be variation of traits within a population, and this variation is going to be caused by mutations. So different alleles would be those variants of a trait that we're talking about. Secondly, these traits have to be inherited from one generation to the next. And we just talked about how mutations can be passed along from one generation of multicell organisms to the next generation through meiosis, or in some cases, asexual reproduction. And in organisms like bacteria that are single-celled, the inheritance can occur from one generation to the next through processes like mitosis. Next, there has to be overproduction or some sort of competition between species or between organisms within the population. And so we might find that certain variations of traits are going to allow for organisms to be more or less competitive. And that actually takes us into our next principle, which is fitness. Organisms in natural selection are defined by their fitness. And again, fitness is based on both the ability of an organism to survive, but also its reproduction. So for example, if an organism lives to be 50 years old, it has a long survival time, but if it doesn't reproduce, then it's not biologically fit. Instead, if an organism lives to be about 25, but produces 5 to 20 offspring, as we might find in the case of some mammals other than humans, then we consider those organisms to be fit because they are passing along their genes to the next generation. And so it's a combination of survival and reproduction that is going to be selected for in natural selection. And in part, this fitness then is then driven by adaptations or these particular traits. And we will talk about some adaptations that affect fitness, but just to kind of give you a brief overview, examples of adaptations that can affect fitness would include camouflage for organisms that are hunted by predators, mimicry of organisms where they look like predators, and even other sexual adaptations. For example, the coloration of peacock males, for example, the beautiful blue and teals that we see in their tail feathers is a form of sexual selection. That vibrancy that we see suggests to females that these males have good health. And so that can be an adaptation that also will affect the fitness of the organism. Now, evolution in part is dependent on gene flow or the lack of gene flow. And gene flow are new variations of genes that are entering or leaving a population. Examples of gene flow can include immigration and emigration, or migration, so new genes moving in or out of a population depending on the introduction of new organisms or the removal of organisms from a population. And also isolation can affect gene flow as well. Let's take a look at an example. Let's imagine that we have an island that once upon a time was a single island in the ocean. But then, by some catastrophic effect, this island has split into 3 various parts. Well, depending on the geographic location of organisms on this island before they split, and depending on the environmental factors and the selective pressures that exist on each of those islands, what we might find is that there would be a disruption to gene flow. So, for example, the organisms on the far left, those shown in orange, would not be able to cross the ocean and share genes or exchange genes with those in purple. Those in purple may not necessarily exchange genes with those in blue, and vice versa. And so the presence or the absence of gene flow, as is shown in this example here, can have an effect on the evolutionary history and the evolutionary development of a species. Another mechanism of evolution is known as genetic drift. Genetic drift occurs when changes in gene frequency are caused by chance. So let's take a look at an example first, and then we'll talk about a couple of more specific cases. So let's imagine here we have bunnies in a forest. Each bunny is represented by one of these circular-like objects. And we have some white bunnies and some black bunnies. And let's suppose that then a storm occurs and this tree ends up falling and killing these 4 poor bunnies right here. Well, what's that going to do? Well, it's going to alter the frequency of black and white bunnies. And so what we would have is then a population that looks like this. And so whereas we began with a population where there was 50% white and 50% black bunnies, we now have a population where there's 16% white and 84% black. There's no sort of selection by the environment on this one as we see in natural selection. Instead, this is by random chance. The bunnies were just in the wrong location at the wrong time. Now, there are 2 specific types of genetic drift that we talk about, one that's called founder effect and the other that's called the bottleneck effect. An example of the founder effect is found in the Amish population and the Mennonite population in the United States. What we find in these populations is that there's a greater frequency of having 6 fingers or 6 toes than there is in the general world's population of humans. And the reason for this is that individuals that are part of the Amish community, just by chance at the beginning of these communities, had a higher case or frequency of polydactylism, as it's called. And so over time, what we found is that this gene has been, the allele for this gene of 6 fingers or 6 toes has been passed along from one generation to the next. Again, without any sort of environmental selection of that trait. In the bottleneck effect, what we find is that a population might decrease rapidly, and as it's rebounding, what we have is a great reduction in the variation of genes within that population. An example of this would actually be the human population in the 15 and 1600s in Europe. During what was known as the Black Plague or the Black Death, we found hundreds of individuals, actually more on the case of millions of individuals, it is reported, to have died from the Black Plague. This greatly reduced the population size in Europe, and so the genes that would have been passed along to the next generation would have been those of people who survived the plague. And so our bottleneck here is an extreme disease. It greatly reduces the population and the variation within that population. And the result is less variation then of the survivors of that particular disease and their offspring. Let's now take a look at 2 examples of natural selection. We are primarily going to focus on natural selection for our method of or mechanism of evolution, so natural selection and mutation. So in this first example, let's look at bacteria and antibiotic resistance. So here we have a population of bacterial cells. You can notice that most of them have a black marker on the outside, but one of them has a blue marker. And let's use an antibiotic that targets that black marker. Well, in this case then, the majority of that population is going to be killed or inhibited in growing by the antibiotic. In other words, the antibiotic here is our selective pressure. The environment in which the antibiotic exists selects against bacteria that have the black marker by killing them off, and the organism that is most fit is the one with the blue marker. It is able to survive the presence of the antibiotic. Not only is it able to survive, but now it can reproduce. And so this one cell would undergo a process similar to mitosis to divide into 2. Those 2 would divide into 4, 4 into 8, and so on and so forth. Now, it's important to note here, too, that we probably would have some genetic drift in play down the road as well. For example, whatever genes or whatever versions of genes are encoded by this particular bacterium right here are going to become those particular versions of genes that are passed along to future generations. And so this antibiotic is greatly reducing the variation or variety within the population and selecting for a particular trait, but then also allowing for only the associated traits, those that are also found within this bacterium, to be passed along to new generations. Let's take a look at another example. Let's imagine this here is a brown soil, maybe dirt or something like that. And in this particular environment, we have mice of different colors. So we've got some black mice and we've got some tan mice. And you can see pretty easily here that one of these might be a little bit easier to distinguish from the other. So let's imagine that we have a predator in this case, which is some form of bird. And this predator is going to rely on these mice for food and for energy. And so this predator here might be flying overhead, kind of having the same image that we have here. And you might argue that it's more likely going to pick off those that are black in color because they better contrast with the background than those that are a light brown or a tan color. Those particular mice are actually camouflaged, and so they have an adaptation here that makes them more fit for their environment. They're able to survive predation. And so if this predator then picks off those that are black, we have those that survive are these tan colored ones, and maybe then we get new variation where they produce some that are very well blended into the environment, maybe some that are a little bit darker, and lo and behold, maybe at some frequency they produce those with black this trait that gives them a black color fur again. And in this case here then, those that are darker or those that contrast with their background are going to be more likely picked off by the predator. So what both of these examples highlight then are a selection by a particular environment. On the top one, that environmental selection was the antibiotic, and the bottom one, it is the ability of the organisms to be camouflaged by their environment, but also the predator that is going to be eating these particular mice. You could see that in both examples, we had variation in the population. We talked about how the traits were heritable from those that survived to their offspring. We talked about competition here, and really the competition in this case is basically the competition to survive the selective pressure. Also competition for food resources that we did not talk about. And there are adaptations that make one set of organisms more fit. Again, fitness here is the ability to survive and reproduce. So one particular adaptation that makes one group of the population more fit than the other to be able to pass along their genes to the next generation. So in summary, we have talked about 4 mechanisms of evolution, including mutations, natural selection, genetic drift, and genetic flow. Again, these do not work in isolation, but instead we often find them working with each other or at the same time in order to drive evolutionary change within populations of species.